Hello. Good afternoon. Hello. Good afternoon. Anybody please respond. Okay. Let's see fertilization. So the machine. Hello. Good afternoon. Hello. Okay.
Hello, sir. Good afternoon.
Good afternoon. Good afternoon. Okay. So today we discuss the pollination and fertilization. So here the machine and the other scientists in 1899. So working independently were able to prove the steps involved in the fertilization angiosperms. So here depend here the fusion. Here the fusion of the pollen grain means male gamete and the fusion with the egg, that is called as the fertilization. Okay. So fusion can be defined as the merging of two nuclei irrespective of their strains, that is the plus and minus. It is involved in the merge of these two nuclei which are the opposite strain. It means the fertilization. So here the fertilization in angiosperms is called double fertilization and triple fission. This is a special character is present in the angiosperm. So here double fertilization. What is the double fertilization here? Okay. So double fertilization and the triple fission. What is this here? Double fertilization means the two nuclei from the male gamete and fuse with the, that is the egg. Okay, means here the micropolar nucleus plus chalazal nucleus.
Hello.
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 Hello. Hello. Hello. Is it clear? Hello.
Sir, your voice is not audible, sir.
Not audible.
Hello.
Okay. Now okay. Now clear. Hello. Now clear. Hello.
Yes, okay, sir. Please continue.
Huh? Is okay?
Okay, okay, sir. Okay.
Okay. Thank you. Thank you. So here. So here the micropolar nucleus plus chalazal polar nucleus, which are the secondary nucleus. These secondary nucleus plus to fuse one male gametophyte. That is the result as the primary endosperm nucleus. So after the egg nucleus plus another male nucleus, that is the male gametophyte, fused to form a zygote. This is the double fertilization and triple fission is occurred in the angiosperm. So here the second and third are the fertilizations. The entire process is highly complicated and the critical details which are being the review. So the time to time. So the basically the reproduction of the gametophytes has which is reached to climax in the angiosperm. So the male gametophyte is three-celled, okay, and the female one 7 cells where the mature. In this mature time, here the three-celled the male gametophyte and 7 celled in the female gametophyte, which are the great reduction. It is depends on the sporophyte is obviously numerous beginning with the nutrition for the growth and development. So what happened here? The maturation of the seed, which has the potential of the growing into the next generation. So this is to the abundant true for these two generations. So that is the diploid and haploid to the. So next one is the pollen and pistil interaction. Here, whatever may be the pollination vector, the pollen ultimately reaches the stigmatic surfaces. Finally, the pollen reaches the stigmatic surfaces. So it is to imperative that both pollen and stigma remains in their peak vitality. So here, while the pollen's life in short case 3-celled stages, those pollen shed into the 2-celled stage for the longer lifespan. So the stigma of the most species remain it's the vitality for longer duration. So here the structure and the function of the stigma. The function of the stigma. Of the stigma. Of the stigma to the receive pollen gain. Here, main function which is receive the pollen grains and to accept the pollen grain. Okay. So here so set and to allow them to the germinate and to provide the nutrition and guide the pollen to grow. So in addition that they are to regulate the flower metabolism soon after the pollination in bringing about the fruit which is stimulated provided by the pollination. Okay. So here the stigmatic epidermis is highly active layer, the functioning like glandular cells providing the stigmatic to excrete. So here So these extract from food for the insects visit the flowers and pollinating. This is useful for the pollinating. It is the chemical nature in the wet type of the stigma is known as a better condition. In addition to the and been mentioned many types of the polysaccharides, flavonoids, phenols, and other compounds are there. Means reducing sugars also to identify these complex exudates. Okay, so this stigma offers the exact chemical need for the right pollen. The stigma to identify the exact pollen grains. This is the classification. This is the classical example for the pollen stigma interaction. Only suitable pollen grains are to germinate on the stigma within the species. Pollen grains are to germinating. After pollination, the stigmatic lobes close and do not receive other pollen grains. After the pollination, the lobes will be closed and cannot accept the other pollen grains. So the closure is due to the stimulus and exercises by the same type, the need by the pollen. When they are in sufficient number, so the pollen germination, the pollen on the being brought the here pollen germination happen here. So this is the pollen tube growth from the pollen germ pores are there through the germ pores. So the pollen tube will be outcome. So which is the so it is depend on the atmospheric factor like relative humidity, intensity of light, and ambient daytime temperature. As a biological factor like the condition of the stigma coating present on the pollen wall and male gametophyte and development like 2 or 3 celled stage. So it is generally 2 celled pollen are slow 3 celled which are the faster. If the pollen grain here 2 cells are there very slow. If the 3 cells are there, it is very fast. So yet the another factor that is substance the pollen germinate mix of pollen from different plants could perhaps induces a comparatively rate in the germination. So this is the some pollen through the germ pores by the endosmosis. The pollen wall is 2 layered. This is the startide. Each pollen grain is composed of 2 layers are there. There is the outer exine and inner intine is present. The exine is formed from the pollen synthesized prime exine on the tapetum synthesized, which is the sporopollenin, and tapetal degradation material are deposited. The inner intine is the cellulosic similar to any other cell wall. This is being synthesized by the microspore culture. Okay, so this is the if the collagens are not acceptable immediately, the stigmatic cells are produced a lot of cellulose which are occupy the intercellular space in the block path of the pollen tube. It is not interested to generate the pollen, so the stigma to produce the cellulose and log the pollen tube growth. The pollen tube grows for the short length for not being the allowed a smooth way it is blocked. So end of it is growth a matrix of the callus. Okay, this is the rejection which is occur in incompatibility process. So rejection rejections here the pollen incompatibility you can see that. So in incompatibility can be exist in the stigma, style, or ovary, with the genetic being the expressed by the two or more genes are there. So this prophytic incompatibility from the pollen side is due to the deposition of the hepatocyte degradation produced with the long exon, which may be manifested on the stigma. So here, the gametophytic incompatibility leads in the non-acceptance protein present in the intent, which also manifests the after pollination and in imbibition. Okay. So here, so another way it is the manifest such as the tube fails to penetrate the transmitting the tissues as it is incompatible of the sizing and cutinase and pectinase. So this is the most significant role for the stigma leaves to recognize the right pollen. This is truly genetic process. So next one is the pollen tube growth. Okay, pollen tube growth. Here, pollen tube on the emergence grows on the surface of the stigmatic papillae in the creeping manner. So here, pollen tube grows on the stigmatic papillae. The growing region of the tube, it is tip, pollen tip. The growing tube growth zone and evacuation zone and cytoplasm zone. So here, each pollen tube here, growing zone is there, and a evacuation zone is there, and another one, the cytoplasm. So growing zone has the dictosome forming the plasma membrane. Is also endoplasmic reticulum and ribosomes and polysomes and microtubules is also there. This is the zone is to involve the protein synthesis as the pollen excreted in the side and center of the from here. So it has the polysaccharide wall and outer pectic and the inner cellulosic layers are there. So behind this is the cytoplasm zone with the smooth endoplasmic reticulum, mitochondria, and other vessels are there. Vessels means vacuoles are there. So another zone, the second, that is the vacuolar zone, is the cytoplasmic vegetative and generative cells are there. So here, three-celled stages present in the dehiscence. The vesicles allocated here fuse forming the tonoplast. This is followed by the large vesicle with the peripheral cytoplasm. Okay. So here, the generative nucleus splits into first followed by the um, vegetative cell nucleus splits into the followed by the generative cell. This is a movement is supported to balance the delicate pollen tube is facilitated by the microtubule and endoplasmic reticulum. So this is secreting the digestive enzymes, the pollen tube grows between the intercellular space of the transmitting zone. So mainly the calcium and boron are known to induce the metaphorical growth and hence the qualitative growth in the cryptographically guided and the enzymatic qualitative reach the style. Okay. So here mainly the calcium and boron, these compounds, these chemical compounds are to induce the qualitative growth. So next one is the style. Okay. The style here, so like this, the stigma is there and qualitative growth enter into the pollen embryo sac. So here there are the three types of the styles are there, which are open, closed, and half closed. Open, closed, and half closed types are. On the open styles, there are those that have the stylar canal lined by the epithelium, secreting some substances to get into the inner lining of the stylar canal, which is moist. The growth of the pollen tube in the here very smooth. The secretion is the similar in the stability to stigmatic exudates. So which is the closer. The styles as a central core is made up of the transmittant cells, which is the elongated as the responsible to the pollination. In the half-closed styles, through the transmitting the cells are present, they occupy a part of the stylar core. Other types of the cells is being mixed with each. So depending on the type of the style, the pollen tube of the species stores the various types of the enzymes are secreted to help in the path through the stylar region. So here, the removal of another, the anthers from a flower arrested with the growth of the pollen tube in the style. Here, they suggested the stimulating the role of the anthers in losing the stylar transmitting cell. So, for example, here, very tough and broad pollen tubes in Cucurbitaceae and Lythraceae, which are prone to highly enzymatic and destroying cells on their pollen pathway. These growing pollen tubes uses the secretion of the epithelial cells in the open styles, and the products are digested cells in other types of its nutrition. So here, starch and lipids often stored in the intracellular space before pollination are completely used by the pollen tube, which are respiratory substances if the pollens are accepted. So the next one is here. So pollen tube enter into the embryo sac. Pollen tube enter into the embryo sac. So here, the pollen tube reaches the based on the style guided the some chromatophorically. Chemotrophically means the chemical substances are there as the concentration of the sugar, calcium, boron, and some terpenoids are there. Based on these attraction, that will be called as a chemotrophical. So on the reaching the placenta, it is the guided in the rare case the obturator. Obturator. So obturator, this is also to guide the pollen tube growth. But in the major cases, the chemical gradient only. This growth with the electroscopic creeping along the surface of the cells up to the ovule. So as a role, the pollen tube enters the ovule through the micropyle, and it is called as a porogamy. So here porogamy means the pollen tube enters into the microphylla through microphylla. That will be called as a porogamy. So sometimes they are entered into the callagel tissue. That will be called as a callagogamy. Sometimes then enter into the mesogamy. Mesogamy means in the embryo sac, between the embryo sac integument. So finally, reaching the embryo sac, the pollen tube creeps along with the wall of the embryo sac and enters into the region microphylla and end of the sac. So all these enters are also guided chemically, means chemotrophically. In porogamy, the pollen tube grows over the ovular surface till it reaches the micropyle and then down to the embryo to the microphylla tongue. So here, synergists. So synergists, they can play an important role in the pollen tube enter into the embryo sac. So synergists can play a vital role. It plays a vital role in the pollen tube enter into the embryo sac. So it is synergists that secrete the some polysaccharides, mucopolysaccharides are there. So which is water spilling into the slimy substances diffusing out of the filter form appearance, aperture start. So the maximum content of the guiding the chemical is proved to exist in the synergies by the autoradiographic so studies using the C14 compound. So here the filiform aperture. So these filiform apertures used in enter into the synergy. When the synergies are lacking as the plumbago type of the so here egg itself develops the aperture and attachment to on the entering the synergy through the passage of the aperture to the synergy cytoplasm and their growth stop. So here the swollen tip of the pollen tube has the male cytoplasm inside the female cytoplasm, which is the outside. Both cells have content, they are different osmotic pressures on the tube wall, which are the finally brushed open and discharging its content. The penetrating energy is generated simply the pattern and solution of the membrane system. So the energy strains are very rapid and became difficult to observe, understand the microscope. So here what happened? The synergy is So here the synergists the synergists are entered into the pollen tube, entered into the from the synergist, synergist the egg. So next one is the pollen tube discharge and bursting of the pollen tube through the subterranean pores in the content of the pollen tube are discharged into the synergist here. Due to all these activities, the synergy cells are degenerated. In this mass of the degenerating cells, all these are organisms, the pollen tube will discharges remain on the isolated conical mass which are the revealing its identity. So these are the spreading into the synergy cells, which consists of two male cells and small amount of the cytoplasm is also there. So remaining the vegetative cell and degenerating the synergy nucleus are in the dark bodies known as the X bodies. They are so on as the X bodies. So here, the male gametophyte comprise the vegetative cell and two sperms. Two sperm nucleus are present here. So these nucleus are one of the sperm is concentrated to the cytoplasmic stance to the nucleus of the vegetative. So here, the two cells form the generative cell is the connected with each other through the membrane wall. Okay. So here, so on the larger will be more some mitochondria and some few plastids are concentrated on the nuclear vegetable cell. So here, the sperm from the fertilized the egg. So other cell is smaller and has no much connection to vegetative nucleus. It has more plastids and known as the fertilized and polar nucleus. So these polarity exhibited by the generative cell is throughout the differences. So some are the isomorphic cells have been reported in some cases barley. Okay. So what is the mechanism here? Nuclear fusion. What is the mechanism is there? The sperms should develop repelling forces between each other and move apart. This is the enabled because the microtubule. The first male gametes are transported to the membrane, which is of the egg cells. So the male cell membrane merge with the eggs and nucleus is transported by the membrane network to form the female nucleus. It is the merging of the cytoplasm and endoplasmic reticulum. It is evident the male gamete participates as the entire cell with the cytoplasm and not only the male nucleus. In this event, the synergic cytoplasm is not involved as it will be degenerating. The male and female gamete nucleus come to in the contact with each other after the and confluence their cytoplasmic membrane. Here, the outer membrane of the two nuclei merge with each other at the point of the contraction between these some bridges are caused. These bridges between these bridges are wide along with the entire area outer membrane like a zipper. So in this process, some amount of the cytoplasm get included with the publication pushed out there. Confluence gets uncompleted. So this is the followed the preciously in the same way to the two inner membrane and now a nuclear double membrane includes two groups in the nuclear context. From the above method, it is clear that there is no loss of the membrane material and both the parental nuclear membrane goes to the zygotic nuclear membrane. So here, the two nuclear groups inside a double membrane fuse to locus by the locus, and this is process very precious. So Navashin. So here Navashina in 1960, they are proposed three types of the bridge of the nuclear are present. So there are the pre-mitotic and post-mitotic and intermediate state. Here the mitosis refers to the zygote first mitosis in the pre-mitotic type of the fission. Both the nucleus fuse before the one set of the zygotic mitosis. In the post-mitotic types of the fuse, is completely at the outset of the zygotic mitosis. In the intermediate type, there is the incompatible mixing later prophase and where the ensues the state. Here, the gametic fusion of the second male and the central cell also exactly similar, but in the direction of the movement is different. So it is the faster than this stingamy and fuse the primary endosperm nucleus, which is the divides almost immediately, resulting in the formation of the endosperm after fertilization. So zygote shows the initially shrinking of the cell size and comes to the polysaccharide. Polysaccharides walls are there without the plasmodesmata. So these shrinkage is due to the elimination of the vacuoles and cytoplasm became the denser with the abundance of helical polysomes and RNA and some dictosomes also. The synthesize some carbohydrates also present. So the polysaccharides forming the wall are biparental. Both the eggs as well as the polysaccharide vessels are formed from the pollen discharge. So after the zygote is undergoes to some distinct phase before they are undergoes to the embryogenesis. So, so double fertilization is a specific character in the angiosperm. Double fertilization is a specific character. It is ends in the formation of the zygote and endosperm nuclear tissues for the embryogenesis. So here double fertilization. So here, the process involves a great amount of interaction between the pistil and the pollen and pistil interaction. So here, the stigma has a great role to catching and sticking the selective pollen grains which are to germinate. So this is acceptance and rejecting the pollen is genetically controlled by the stigma. So here the okay. So structure of the stigma and chemical composition of the exudates are complex and varied nature on the germination and pollen tube formation, which are the chemotrophically guided from the stigma. Okay. So finally, here they are to form the embryo. Okay. This is the double fertilization and the triple fission. Okay. So here